video we have here these 20 questions to answer on the topic plant and animal nutrition. So this video will be about plant nutrition. So ensure to stay till the end of this video. Let's go ahead and consider the first question here. It says, which of the following structures are characteristics of insect pollinated flowers? We have A. Brightly colored petals and scent nectar. B. Very light, numerous and pendulous stamen. C. Flowers are usually small and inconspicuous. D. Dull colored flowers with no nectar. So guys, the answer to this question is option A here. Brightly colored petals and scent nectar. So this allows them to, the brightly colored petals actually make them more attractive, and as well the scent nectar makes them attract the insects in order to be pollinated by these insects. So that's it for this question, and let's go ahead and consider the next question, guys. That's question number 2 here. It says, which of the following is considered to be the first product of photosynthesis? We have A. Hydroxyl ions, B. Starch, C. Protein, D. Energized chlorophyll. So guys, the answer to this question is option B, and that's starch. So let's go ahead and consider the next question. That's question number 3 here. It says, the following organisms are examples of carnivorous plants except A. Utricularia, B. Pitcher plants, C. Venus flytrap, D. Gloriosa. So guys, the answer to this question is option D, and that's Gloriosa. So the rest here are actually carnivorous plants. The Utricularia, the pitcher plants, the Venus flytrap are all carnivorous plants. But guys, Gloriosa is not a carnivorous plant, and that's the answer to this question. So let's go ahead and consider the next question, guys. That's question number 4 here. It says, at the end of photosynthesis, the green leaf is tested for starch and not glucose because A. Starch is stored only in leaves. B. The glucose is used up immediately to provide energy for the needs of the leaf. C. The glucose formed is immediately converted to starch. D. There are no chemicals for glucose tests in leaves. So guys, the answer to this question is option C here. The glucose formed is immediately converted to starch, and that's it for this question. Let's go ahead and consider the next question, guys. That's question number 5 here. It says, use the information below on factors required during photosynthesis to answer the 2 questions. We have I, CO2, II, chlorophyll, III, water, IV, ATP, V, NADPH. So the factors required for the dark phase of photosynthesis are A. IV and V only, B. III, IV and V, C. III and IV only, D. III and V only. So guys, if you can remember the process of photosynthesis, we have the light phase and the dark phase. So guys, in the dark phase, the materials required are usually ATP and NADPH. So that's it for this question. Option A is the answer to this question. So let's go ahead and consider the next question, guys. That's question number 6 here. It says, the amount of starch in a germination seed decreases because the starch is used up A. for respiration and building up of cells, B. to build cellulose cell wall, C. for the production of enzymes, D. to develop meristematic tissues. So guys, the answer to this question here is option A, and that's for respiration and building up of cells. So guys, let's consider the next question, guys. Question number 7 here. It says, the classes of plants, the root system in diagram I and II below represents respectively are A. melocotyledona and dicotyledona. B. Monocotyledona and monocotyledona. C. Dicotyledona and dicotyledona. D. Dicotyledona and monocotyledona. So guys, the answer to this question is option D here, and that's dicotyledona and monocotyledona. So let's take note that in dicots you have a taproot system, which is as shown here, and then in monocots we have a fibrous root system, which is shown in II. So that's it for this question. Let's go ahead and consider the next question, guys. So the question here says, the diagrams below are illustration of an experimental setup to demonstrate a type of trophic response in plants. Study them and answer the question below. So the question here says, the conclusion drawn from the experiment is that A. Shoots of the plants are negatively thigmotropic. B. Leaves of plants are positively phototropic. C. Shoots of plants are negatively geotropic. D. Roots of plants are positively hydrotropic. So guys, the answer to this question here is option D. Roots of plants are positively hydrotropic. So that's it for this question, guys. Let's go ahead and consider the next question. Question number 9, it says, which of the following pairs of organisms are photosynthetic? We have A. Volvos and rhizopus. B. Amoeba and paramecium. C. Nostoc and plasmodium. D. Chlamydomonas and euglena. So guys, the answer to this question is option D, and that's Chlamydomonas and euglena. So that's it for this question. Let's go ahead and consider the next question, guys. That's question number 10 here. It says, the diagram below is the root system of a leguminous plant. It says here, the structure labeled II are A. Root nodules. B. Underground root nodes. C. Groundnut seeds. And D. Root tubers. So guys, the answer to this question here is option A, and that's root nodules. So the parts labeled II here are the root nodules of these leguminous plants. So that's it for this question. Let's go ahead and consider the next question, guys. Question number 11 here, it says, which of the following types of placentation is not common among Synacarpous pistils? We have A. Parietal, B. Marginal, C. Axial, and D. Free central. So guys, the answer to this question is option C here, and that's axial placentation. So that's it for this question. Let's go ahead and consider the next question. So here we also have a diagram, and it says here, study the diagram below. So the question here says, which of the following structure in the diagram are involved in the entry of carbon dioxide for photosynthesis? So we have options A, I, III, and V. B, I, and VI. C, I, and III only. D, I, V, and VI. So guys, the structures in this diagram that are involved in entry of carbon dioxide for photosynthesis will include I, V, and VI. So that's option D here. And that's it for this question. Let's go ahead and consider the next question, guys. That's question number 13 here. It says, conversion of glucose to starch in the leaf during the day, principally A, enables glucose to be used up. B, enables photosynthesis. C, prevents osmotic problems. D, enables the leaf to store the starch. So guys, the answer to this question is option D. It enables the leaves to store the starch. So that's it for this question. Let's go ahead and consider the next question, guys. That's question number 14 here, and we also have a diagram here. It says use the diagram below to answer the question, and the question here says the part labeled II is the A, spongy cell, B, stoma, C, palisade cell, D, guard cell. So guys, the answer to this question is option B, and that's the stomach. So let's go ahead and consider the next question, guys. That's question number 15 here. It says, which of the following statements is not true of the light stage of photosynthesis? We have A. Chlorophyll is energized by sunlight. B. Carbon dioxide is produced by hydrogen atoms in NADPH. And then C. Water is formed. D. Water molecules split into hydrogen and hydroxyl ions. So guys, the answer to this question is option B here, and that's carbon dioxide is reduced by hydrogen atoms in NADPH. So this is not correct for the light stage of photosynthesis, and this does not occur in the light stage of photosynthesis. So that's it for this question. Let's go ahead and consider the next question. Question number 16, it says here, which of the following bacteria lives in the root nodules of leguminous plants? We have A. Rhizobium, B. Nitrobacter, C. Azobacter, and D. Nitrosomonas. So guys, the answer to this question is option A, and that's Rhizobium. So that's it for this question. Let's go ahead and consider the next question, guys. It's question number 17 here, and it says, the steps below are involved in the testing of a leaf for starch. Study them carefully and answer this question. We have I. Decolorized leaf. II. Deep colorized leaf in warm water. III. Place leaf in boiling water. IV. Add iodine solution. The best time to pluck the leaf for the experiment is A. 3 hours after sunset. B. 6 hours after sunset. C. After being exposed to sunlight. D. Before sunrise. So guys, the answer to this question is option C, and that's after being exposed to sunlight. So that's it for this question. Let's go ahead and consider the next question, guys. That's question number 18 here. It says, a solution which contains all the required elements in their correct proportion is known as A. Complete culture solution. B. Culture nutrients. C. Growth medium. D. Nutrient solution. So guys, the answer to this question is option A, and that's complete culture solution. So let's go ahead and consider the next question, guys. That's question number 19 here. It says, the plant cell mostly stores as A. Starch, B. Auxins, C. Hormones, D. Glycogen. So guys, the plant cells actually store food as starch, and that's option A here. So let's go ahead and consider the next question, guys. That's the last one for today. Question number 20 here. It says, the deficiency of manganese in plants results in A. Poor root development, B. Death of shoots, C. Leaves turning yellow, and D. Stunted growth. So guys, the answer to this question is option D, and that's stunted growth. The deficiency of manganese in plants will result in stunted growth. And that's it for this question. So guys, this is where we end this video for today. If you enjoyed this lesson, please give it a thumbs up. Don't forget to subscribe to this channel if you're not a subscriber. Also, if you have any specific question regarding this video, you can drop your question on the comment section below, and we'll ensure to give you a response. So finally, guys, we want to encourage you to join our YouTube channel community by hitting the join button below, and we'll see you right there. So thank you once again for watching this video, and see you on the next one.